Welcome to Biology Made Easy. Today we are solving genetic questions for AS or A-level students. Today the questions is on linkage, chiasmata and crossing over, and looking for crossing over values. Welcome. The question is, in maize plants, the genes for colored seed and full seed are dominant to the genes for colorless seed and shrunken seed. Pure breeding strains of the double dominant variety were crossed with the double recessive variety, and a test cross of the F1 generation produced the following result. Colored full seed 380, colored shrunken seed 14, colorless full seed 10, colorless shrunken seed 396. Questions. A. Explain and state the ratio of phenotypes in a dihybrid test cross as the test above. B. Explain the discrepancy between the expected results in A and the results stated. C. Calculate the crossover value or crossover frequency. D. Calculate the distance in units between the genes for colored seed and seed shape on the chromosome. E. Using annotated diagrams, explain chiasmata and crossing over between genes for colored seed and seed shape. Well, we need to analyze the data and solve the problem. In maize plants, genes for colored seed, so we know we are going to deal with colored seed, and full seed are dominant, so we are also dealing with full seed, and we are told that is dominant. So if we give colored seed A and full seed B, capital A, capital B, they are dominant to genes for colorless seed, so here is colorless seed. So colorless seed will be small a because colored seed is dominant, and shrunken seed is here. Also, shrunken seed will be small b. Pure breeding strains of the double dominant. Pure breeding, we have big A, big A, big B, big B. Pure breeding strains of the dominant variety, that's it. Pure breeding homozygous dominant variety were crossed with double recessive. We have this cross, double recessive is this one, and a test cross of the F1. So the F1 will have to come in. We have the F1 here, which will be big A, big. That will be the F1. Test cross of the F1 means you cross the F1 with a double recessive. That's a test cross. And this test cross, we are told, yielded these results. Question A says, explain and state the ratio of phenotypes in a dihybrid test cross as the test above. So it means that if these two genes, if it's a dihybrid with no linkage, will these results, and you know that if a test cross of this nature is done and there's no linkage, we should have the ratio 1:1:1. If this test cross is done and there's no linkage, we should have gametes as this, written out like this. This one is the gamete here. We put the male gametes here, Punnett square, the female gametes here. This is what we get. Colored full seed, colored shrunken seed, colorless full seed, colorless shrunken seed. And we have 1:1:1:1. 1 out of 4. So the ratio should be 1:1:1:1. So if this test is done in a dihybrid, no linkage, we should have the ratio 1:1:1. But look here, colored seed, what we had was 380, colored shrunken seed 14, colorless full seed is 10, colorless shrunken seed is 396. These are the results here. And these results, if it's a dihybrid test, you will not get this result. You get 1:1:1. That is here. So in question B, we are supposed to explain the discrepancy between the expected results in A and the results stated. The expected results in A, which we have proved, is that we should get a ratio of 1:1:1:1, meaning colored full seed should be all these should have almost the same numbers. But if we looked at what we got, the discrepancy is that the parentals colored full seed is 380, colored shrunken seed is 391. These two parentals seem to be 1:1 ratio, and the recombinants 1410 is almost 1:1. There's linkage between the genes for seed color, that is A, and seed shape. The two genes are on the same chromosome. They are not on different chromosomes as we have stated here, where this is on one chromosome, this is on one chromosome, all right? The two genes are on the same chromosome, linkage. But 24, you see the recombinants are 24? 24 out of the total is 800. The recombinance is 24, the total is 800. 24 out of this total 800, the genes for seed color and seed shape, there was crossing over between the genes. And that is how come the recombinants showed. And in the rest, that is this and this is 7,7Cs. The rest of the seeds, that is 7,7C seeds, the genes for seed color and seed shape remained linked. That is why we have this results instead of 1:1:1:1. Is the linkage. So we have explained B. Question C says calculate the crossover value of frequency. And what we have said is the genes for seed color and seed shape are on the same chromosome. Seed color, seed shape. And yet there was crossover between the genes for seed color and seed shape in 24 out of the 800 Cs. There was crossover. And that is how come we had this. So we are being told to find out the crossover value, the frequency with which the crossover took place. That is it. The crossover value. This is the equation that is useful. All right. So the crossover value, you have the number of recombinants over the total number of offspring times 100%. The number of recombinants is 24. The total number of offspring is 800 times 100% will give us 3%. So that's the crossover value. Now, the next question is to calculate the distance in units between the genes for colored seed and seed shape on the chromosome. Good. Simply, what we are being asked is that we have said that there's crossover between these genes. If this is the chromosome, we should determine the distance between this gene and this gene on the chromosome. And the crossover value is what is used to calculate the distance between this and this genes. The crossover value of 3% means there's 3 units distance between gene A and gene B on the chromosome. The crossover value is used as the relative positions of genes on the chromosome. Good. So that's the crossover value and the distance between genes. Related, they are always related to the crossover value. Now the last question, using annotated diagrams, explain chiasmata and crossing over between genes for colored seed and seed shape on the chromosomes. We have ascertained that colored seed and seed shape, they are linked. They are on the same chromosome. So for this individual, has this chromosome with this gene and here this gene, we have this for colored, this for shape of the seed, and its homologue is the small a and small b. So we start with homologous chromosome like this, and during meiosis, prophase, we see the chromosomes as 2 sister chromatids held by a centromere. So this one chromatid, held by a centromere. That's this chromosome. This chromosome also will appear as 2 sister chromatids held by a centromere. Then chiasmata formation, that is these 2 non-sister chromatids coil around each other and break and then exchange genes. And that's crossing over. So this one we'll end up having gene A here with gene small b here. And this one we're having gene small a here and having gene big B here. So crossing over takes place, the exchange of genes between non-sister chromatids. Very important point. Then the centromere breaks and you have this chromatid, chromatid, chromosome, this one and this one. So in the final analysis, you end up by having these two as recombinant gametes. And this is a parental gamete, this is also a parental gamete. So finally, this is what happened during chiasmata and crossing over, which yielded these recombinants. And the chiasmata and crossing over did not take place in all the seeds, and it took place in 24 out of the 800, but in this parental, which is 776, the genes remain linked. That is the explanation of today's question. If this has been useful, please give us your likes and kindly do subscribe. Thank you very much and goodbye.